My name is Calum Walsh. I'm a postdoctoral researcher here at the Centre for Pathogenomics in the University of Melbourne, and I'm going to be talking to you about a human microbiome biobank that we've been putting together and what we're using it for. So just as a quick background, this is the Centre for Pathogen Genomics at the Peter Doherty Institute. We're primarily a group of researchers and public health experts that work in using genomics to study human pathogens. We are embedded within the public health lab in the state of Victoria, but we also have, so we do a lot of collaborations with other public health labs in countries around the Asia Pacific, trying to get helping them use genomics and bioinformatics analysis in their public health work. But we also have a strong research component where we have all these different research teams, and I lead the microbiome theme along with Dr. Glenn Carter. So I'm a microbiome bioinformatician, while he's more of a classical microbiologist who's an expert more in the lab work and animal models. And together we've been putting together this microbiome culture collection where to date we've isolated over 2,000 bacteria from 130 different species from healthy human fecal samples. And we routinely do Illumina whole genome sequencing on these and do a lot of in vivo and in vitro characterization to try and get an understanding of what they are and what they're doing. So we routinely do relatively simple stuff like phylogenetic analysis and pangenome reconstruction on the species level. And then for every isolate we have, we do a bit of work kind of more tailored towards the gut microbiome, where we're looking at the primary metabolite pathways using software like GutSmash, secondary metabolites such as antimicrobial peptides using AntiSmash and Bagel. And then we're trying to do genome scale metabolic modeling on these isolates that we're constantly trying to refine using in vitro tests, in vitro assays and characterization and metabolomics to try and improve these models. And we also do a bit of safety screening, just looking for AMR genes and virulence factors, etc. A lot of the work for this biobank was done by a PhD student called Charmaine Tan, who's been looking at non-antibiotic based methods to kill carbapenem-resistant Klebsiella pneumoniae, which is a gut colonizer and an important human pathogen that kind of persists in the gut at low levels and can cause human disease and outbreaks when the conditions are right. And what she's been doing is she developed this miniaturized high throughput co-culture assay where she co-cultures the CRE-Kp along with each of our different commensals in a 96 well plate format and grows them over a period of time, and then does plating to enumerate the CRE-Kp after the co-culture. And in the bar chart down here on the bottom right, you can see the black line is our CRE-Kp control, and the blue bars are all of the isolates that have a more than 1 log reduction in CRE-Kp levels during co-culture. And then on the bottom left here, we have 53 isolates that completely kills Kleb in these triplicate assays. So we're really interested in those. So Charmaine has done a bit more characterization on these, doing stuff like colony overlays to see if they are able to kill Kleb directly by producing antimicrobials, or else looking at this nutrient blocking potential. So is the commensal able to use some of the same nutrients that Kleb is using? And therefore that can be why it could outcompete it in the co-culture assay and might be potentially why it would be able to outcompete it in the gut. So there was a paper published in Science at the end of 2023 where a group developed a method to kind of quickly try to infer this nutrient blocking potential based on functional overlap, where you compare the two genomes of your MDRO and your gut commensal, and just by looking at the percentage of shared proteins, you get a really good estimate of how likely the commensal is to be able to outcompete the MDRO. So we've been developing methods to try and refine this, to speed it up and to make it a bit more accurate by weighting specific features that we think are more important based on in vitro testing and the literature. So we can add different weights to different features using our algorithms, and we can rank our isolates from 0 to 1 based on how well we predict they would kill Kleb pneumo in vitro or in vivo. And we've also developed methods to kind of speed up the process of this, so we can test a bunch of different strains in a consortium or in a small defined community really quickly. So we can make artificial communities of, here I'm showing between 3 and 15 strains, but we've gone all the way up to 50 to make these really complex communities, and we can predict how well we think they'll be able to outcompete and decolonize Klebsiella. And Shemaine has done a bunch of testing on these, and the results are pretty good. So all of these here, these are 9 different communities of size 3, 5, or 8. So that's the number of strains that are in the community. And by testing these all had the same predicted anti-CRE-Kp activity. And when we co-cultured them with Kleb, we got the same between 4 and 5 log reduction in Kleb pneumo growth. So it's kind of panning out where the predicted activity actually is relatively similar to what we're actually seeing in vitro. But she's kind of gone a bit further, and she's starting to look at the strain-specific responses or the strain-specific effect. So this example, we have a community with 3 species that gives this 5 log reduction in CRE-Kp when they're co-cultured. But by swapping in different strains of the same species, we can see a differential effect. So some strains cause a larger reduction in CRE-Kp than others. And this strain here, which we're calling delta, by including this in the community, it completely wipes out CRE-Kp in our in vitro models, much more than it did when it was co-cultured with CRE-Kp on its own. So the background consortium is kind of enhancing the anti-Kleb effects of this strain. And we can refine these and add this back into our models to try and better predict the communities. Another one I'm going to talk about is an experiment performed by a researcher here in Lucy, who has been working on this this model of MDRO gut colonization, where mice are pre-treated with piptaz, the antibiotic, for 2 weeks to disrupt the gut microbiome, and then they're colonized by CRE-Kp, and then they're either left untreated as a control, or else we feed them these communities that we've rationally designed using our algorithms, communities of different sizes, so between 3 and 15 different strains in a consortium together to see if they can outcompete CRE-Kp and clear it. And interestingly, we saw that our largest community with 15 distinct strains outcompeted Kleb and cleared it within about 11 days. But our simplest community with only 3 different strains cleared it in about 16 days. So this was quite promising. But actually, after 11 days, 80% of the mice in the group that were fed the 3-strain cocktail completely cleared it. But there were a couple of mice that still had high levels of CRE. So we kind of had this responder, non-responder effect. So we're doing metagenomics now on all the fecal samples that we've collected from this trial, and we're going to try and look into this to see what's different about the microbiome between the responders versus non-responders, looking at how well these strains engraft and whether they support each other's growth, to try and get a bit more information so we can refine our algorithms and our models for predicting these important consortia. And just finally, we're going to mention that our culture collection is public and we are open to collaboration. We want people to try and use these strains in their own work. So Charmaine has put together a Shiny app that's available online where people can query our database and look at the different species we have, filter them by different features. So if you wanted the Streptococcus aureus to produce as an antimicrobial peptide, you could filter our database and see if we have any strains that match that criteria, and you could request it, or we could send you the genome sequence for your own analysis. And our plan in the short term is to get this integrated into our Center for Pathogen Genomics bioinformatics portal, which has been put together by a postdoc in our group called Waitama, where he's got about 40 or 50 different publicly available tools that are running on our servers here in Melbourne, but you could upload your own data or use public data and interact with them through a web browser interface to make it really easy to run. And our plan is to make all of our genomes easily integratable into this, so you can just pull in strain IDs based on their genomes or based on their features and integrate it into some kind of genomic analysis that you want to do through this web interface, and see if it's worth getting in contact with us to see if you could access that strain. And then finally, just as a thank you, these are all the people that did all the work in this. Shemaine, Nick, and Taylor did all the work isolating all of the bugs and characterizing them and doing the genome sequencing and putting together a database. Glenn and Lucy did all the lab work, and then my bosses, Tim and Ben, who lead the group and lead the research. So thank you very much for listening, and thank you for the organizers for inviting me.